Microwave Technologies, which is a micro Edinburgh-based SME. We have developed and manufacture a method of heating fluids on a continuous basis. We call this method microwave volumetric heating. And what we do is we can take conventional microwaves and they're placed around. Microwaves are the magnetrons are the green boxes, and they deliver energy into the flowing stream of fluids, which can be liquids, suspension, or semi-solid material. So we use the flow rate to control the temperature of the liquid. The Teflon tube remains at the same temperature as the contents of the tube, so you don't have burn-on issues. There's a number of technical benefits associated with our technology. We have two machines shown here that are in operation in Scotland. On the far left is a 21 magnetron machine which can produce about a tonne of cooked product an hour. And then you might have heard of Get Juiced, it was in the press just before Christmas, who adopted our technology, and they use the technique to pasteurise fruit juice. They literally take the oranges, squeeze them, and then produce an extended shelf life product that retains all its nutrients. So our technology is scalable. You can have a machine from 3 magnetrons up to, well, as many as you want. It's modular, it's microwave volumetric, and unit is here, so it can fit into a complete process line. So we've used the TSB funding. We had 2 feasibility studies in the last Nutrition for Life, and we've used it to validate some of the technical claims and to control our IP. So we're in control of the projects we've done. On the top right is the slide is a microbial slide is of Mycobacterium avium paratuberculosis, which is present in milk. And we've been able to show with our technology that we can reduce the microbes about 10 to 15 degrees lower temperatures than conventional methods of thermal processing. So we've used that to extend the shelf life of products and increase the biosecurity of food products. As I mentioned, Get Juiced have been using the machine to flash pasteurise fruit juice, and they've shown that there's retention of vitamin C and also the flavours and colours of the fresh produce. So you can put the juice in, and taste tests have been able to not tell the difference between the raw product and the processed product. And then our other feasibility study was with Queen Margaret University, where we looked at salt reduction in the products. We've noticed that with our method of heating compared to conventional processing methods, that you're able to reduce the salt because our product tasted a lot saltier. So we were able to show that for certain products like cheese sauce, haggis, and black pudding, we're able to effect about 15% salt reduction, and that's without compromising flavour and shelf life. So we're looking for collaborative partners that have expertise in nutrition and microbiological expertise. We're also very interested in the economics of food production and processing, and where our technology can add benefits to other processing methods. And we're also looking for industry partners. We're particularly interested in applications with the dairy industry and the fruit and vegetable industry. So I think that summarises where we want to be. Okay, thank you very much.
My name's Cindy Ledgeworth, and I'm a medical herbalist. I've recently retrained. Originally I started my field of research as an undergraduate in psychology and neuroscience. I currently blend exquisite hand-blended herbal teas. My teas are loose so that the integrity of the herbs aren't lost and the volatile oils aren't lost whilst during packaging and processing. So, for example, I have a loose leaf peppermint, which when you drink it, you can almost immediately feel the effects on your stomach, on your sinuses, and through your head. So I'm looking in regards to other herbal teas which are out there. Currently, the majority, through convenience, are packed into tea bags. By packing the teas into tea bags, more often than not, you do lose many of the nutritional properties within the teas. Over the last 5 years, there's been a huge influx of herbal teas on the market, also loose black teas. People are looking towards quality, they're looking towards taste, they're wanting to know what is in their product, what they're eating. I'm finding that I have a group of, for example, 16 to 25-year-old men who are coming to me to look for the nutritional benefits of the teas. They're liking hibiscus, which obviously lowers blood pressure, is antioxidant, it's high in other nutritional elements. So with the herbal teas, they're caffeine-free, there's no sugar, there's obviously no salt, they're nutritious, and they're available for all. I have mothers coming to me with their young children who are wanting to take the sleep tea and knowing the dose so that they can help their small ones get off to sleep without having to resort to the traditional cough medicine or even brandy. It's true. Some of my areas of expertise are that I'm able to, through the therapeutic knowledge of traditional research and also through pharmacological research, is understand the benefits that these teas bring to people. However, much as with the herbs, this is all based on hearsay rather than empirical evidence knowledge, which is what happens in the pharmaceutical industry. Some examples of my blends are energy tea with ginseng, immune tea with echinacea. As we know, in the last year, the government have brought out guidelines on echinacea use in those children under the age of 2 years old. So these are the areas where I'm looking for research with the herbs that were able to substantiate the medical properties, evidence, and health-bringing properties of those herbs and plant systems used. Memory tea with ginkgo biloba. Over the recent years, there's been a lot of research of ginkgo biloba in the use in Alzheimer's. However, there isn't research in ingesting ginkgo in a liquid form rather than as a leaf, incorporated either as a tablet or a brewed stuff. I'm currently struggling with finding out any research of where we're looking at herbs which have been ingested through herbal preparations such as tea. Tincture, we've got empirical research and there's some pharmacological research, but again, it's very much lacking. There's other ones which are more ethical. So, for example, love and courage, borage and rose. Borage has traditionally been used to promote courage. Back in the Victorian century, ladies used to give borage to lovers so that they would have the courage to give them the proposal that they're looking for. Borage does and is known to work on the adrenal glands. However, many of these herbs have gone out of use, but people are looking for alternative medicines and alternative ways to treat themselves and also different things to drink. Caffeine is now seemingly on the way out, as are nutrient drinks such as sugars and also those which are sweetened, again with a possible feasible link with diabetes. A correctly, adequately blended herb will be sweet, it will be spicy, it will give all those desired effects patch-free, which also work not only on the taste but physiologically as well. So I currently import my herbs from abroad. I've found that the herbs that we generally supply in the UK, and I was originally organic certified, but have put that to the side, but the herbs I'm importing not only have a better packaging delivery system, they come to me in a better shape and form, but also they're stronger. So, in regards to some of the areas which I can offer, I have both medicinal and therapeutic knowledge of herbs. I'm particularly skilled in blending herbs for both quality taste and nutritional benefits and understanding the physiological effects of those herbs as well. Seeking collaborators for, as much as the speaker has been about, is research into the herbs, whether or not it's by participant trials or by testing the products. So, for example, I'm looking for a seaweed tea, which will work on the thyroid system. Again, how much thyroid, how much iodine is coming out of the seaweed? Does it make it a viable potency for people to ingest for it to resolve certain middle-age problems, so to speak? Also, looking for help from the food and drink industry sector. Thank you.
My name is Madalina Nakshu. I'm from Delegate Institute, which is a part of our University of Edinburgh. A main area of research in the Delegate Institute is food security, which implies sustainable food sources. I would like to speak a bit of a showcase of a project which is which is currently running in the Rowett Institute, which helps me to underline the expertise of our team. The project is entitled Healthy Parts of Sustainable Ingredient Selection in Food and Drink Industry. This is a strategic partnership funded by Scottish Government, which looks to alternative sources rich in proteins to partially replace meat-based diets only. Here we look for locally grown sources rich in protein like beans, pulses, or some grains, and we offer our expertise is in a comprehensive food analysis. We are doing macro, micro, and non-nutrients analysis. We are working with last generation techniques like HPLC-MS, GCMS, ICP-MS, and in order to prove the health effects and health impacts of novel foods such as protein isolates or rich plantains or alternative foods, we have a vast experience in human dietary intervention. We do from acute to chronic studies where we look from the postprandial impacts of these foods up to the fate of macromolecules such as protein and phytochemicals to the systemic and the colonic level. We also have expertise in consumer studies where we try to understand potential barriers of acceptance of novel foods from the customer up to the industry level. Shortly, I'm going to present our team. The project is led by Alexandra Johnson. Her area of expertise is food formulation for enhancing of satiety. Dr. Wendy Russell, she's overseeing the plant products. Her area of expertise is bioactivity metabolism of diet components. Myself, I'm Madeleine Action. I'm overseeing the human study. My area of expertise is natural compound-based products with biological activity and impact on health. We have Dr. Sandra Carlyle, which is overseeing the consumer studies. She's a social anthropologist. Her area of expertise is food choices through Scottish society. And we have Dr. Alan Rowe, which he's in charge of all the industrial inquiries of the related research. Thank you very much.
My former name. I'm an agroecologist, so I'm not a food nutritionist at all. And I'm here representing the James Hutton Institute. I could talk about wild plants, which is really my main love. I look at within species diversity of wild plants. I've got some collaborators in the room already. But I'm going to talk about grain legumes. In particular, I think a lot of what I'm going to talk about is to extend the value to an existing TSB-supported project, which is the Sustainable Protein Production Call. Our coordinator sitting over there. But what I'd like to talk to you about is a little bit just about the biology and ecology. And when I talk about nutrition for life, I don't just mean nutrition for our life. I think there are ways of cropping that can make nutrition benefit the environment as well as people. I like to use legumes as one of those examples. We've just had a taste of that from the previous speaker. Legume production in Europe occupies a quarter of its acreage compared to the 1960s. And this contrasts with legume production in many other countries that have realised the environmental benefits. High protein production in countries outside the UK have been realised in several ways. They've recognised the economic and environmental costs of not using legumes. They've matched specific plant lines, specific varieties with effective strains of legumes are important for those non-plant people here. They don't need fertiliser. They get their fertiliser from the air. They don't require fossil fuel-derived fertiliser. And that's a combination of a symbiosis with a bacteria that we call rhizobia. They match specific varieties of rhizobia. And that, of course, can affect the nutritional quality of that legume. Not a lot is known about that. And also, of course, they've tied legume production to animal and human well-being. Culturally, legumes are not yet as widely accepted in this country as they are in others. Of all the grain legumes, I could talk about a few. Fava bean is the most promising. There are, of course, in Britain we can grow lupins, we can grow peas. Fava beans is something very special about its functional attributes. It can fix very large quantities of nitrogen, much more than peas. Of the amounts it fixes, more is left in the system. A lot of the problems we have with agricultural systems today is that too much is removed from the system. Fava beans leave nutrients in the system, reducing fertiliser requirements, not just for this crop, but for the crops that are sown after it. It has very deep tap roots. The nitrogen and carbon it deposits through the whole of the soil profile, not just the top layer. It also supports farmland biodiversity, including some insects and pests. So if I was to say what are the two main objectives to realising high-value green legume foodstuffs, optimising the agronomy is obviously one thing, but and this is why I'm here. The second one is developing premium products, and it's one of the waste products from the TSP. I say waste. We're using a product that's not being used to feed fish. It's being used to feed poultry and pigs. It could be used that waste product, that product's going to feed animals, could be used for many other things. I'm happy to talk about that later. But what can JHI offer? We have a large germplasm collection. We have breeding lines. We have assessed quality traits in a lot of these already. We have some experience in other legumes. We have a wild legume collection that's particularly interesting. We now have collections of rhizobia isolates. We can do a very large range of analysis, and I'm sure my colleague Derek Stewart can talk about many of the other things that we can do when he speaks later. So for the moment, what I haven't mentioned here is my weeds. We have, with collaborators at Strathclyde and also collaborators at the University of Aberdeen, been developing some of the byproducts of the existing TSB project for beer production, for bread making, and also we've got some novel products from wild plants that are close to patent, but we need to trial these for their in vivo efficacy. We know how good they are in the test tube. We don't know how good they are when they're ingested. So I'll leave that there. Thank you.
We're talking here about novel methods for authentication and traceability, and I'm failing miserably. This is about safety, authenticity and traceability, and we were onto this issue even before we were getting peckish watching the Grand National. We've been working in this area for quite a long time. At the moment we can do this kind of thing with DNA. We can get species and individuals. You can buy tests online these days for things like horse DNA. But what we are trying to do is take this a bit further. We're convinced that there's a need for better methods of authentication and traceability. We use two techniques for this. One is natural abundance variations, and the other one is epigenetics. First of all, the epigenetics, it comes from the Greek meaning epi for upon or over, so it's information that's laid upon the genetic information. And it's basically a record of life. It's how life impacts on DNA. And there are things like methylation sites that on DNA and histones, etc., that accumulate over time. So it's a way of getting it history, if you like. Natural abundance variations, I'll explain a bit more. Basically, you can do the same thing, species and individuals. You can do biological, geographical origin, things like following food chains, working out ages of species that are in food, looking at additives, processing history. So it's following the food chain, basically. The natural abundance work is based on the fact that there are natural variations in isotopes throughout the world. So these are stable isotopes. These are not dangerous radioactive isotopes. They vary between individuals. and you can actually use this variation to find out where things came from. Now, there's been some work done on this, looking at usually total amounts of isotopes like carbon-13, oxygen-18, etc. But what we do is we add many more dimensions to that information by looking at a wide range of components of food and a wide range of isotopes as well. So it gives us the additional dimensional information which allows us to say what is unique about certain components in food. The epigenetics is the same kind of concept. So what we've been doing is applying this to foodstuffs. For example, if you're interested in whether your salmon was fed vegetable oils or fish oils, we can tell that kind of thing from the variations in the natural abundance. We'll be looking at meat products, etc., and coming up with lots of information on that. So basically, we have these technologies, these techniques, where we can measure these things with gas chromatography, combustion isotope ratio mass spectrometry, if you're interested in for the mass spectrometry, natural abundance. For the epigenetics, pyrosequencing, array technology, etc. So we have all this experience. What we want to do now is we want to point it at some real problems. We want to work with people that have real life issues about traceability, working out what components there are in foods, and we want to take this technology and apply that. And I think I'll leave it on a yellow. Thank you.
I'm here to represent the Morden Group. We're located just outside Edinburgh, and our focus is on livestock health solutions. Our parent organisation is a charity, the Morden Foundation, which has 10,000 members from the farming and veterinary communities, so we're very much embedded within that community. Within the group, the largest company is the Morden Research Institute, and within the institute there's nearly 200 staff who have a wide range of scientific expertise across bacteriology, virology, parasitology, epidemiology. We work with a number of different livestock species, cattle, sheep, pigs, poultry, and more recently we're becoming involved in fish vaccines. The outputs are vaccines, diagnostics and disease control measures. And we have very much an applied focus which builds on the basic research that goes on at the Institute. Another company within our group is an SME, Morden Scientific, which I'm involved in, and we do contract research and testing services. The facilities that we have are, we can work to containment levels of up to category 3 for both animal studies, laboratory work, and we work with large animal systems as well as smaller animal models as well. We have established quality systems, so depending on the nature of the work that needs to be carried out. We have experience with the Nutrition for Life call in that one of our scientists, Karen, who's in the audience, was involved with AMT, who spoke earlier on the para-TB work in milk, and that project went well, and we're interested in becoming involved in future projects. So our fit with today's call is that we could be a potential collaborative partner in the area of food safety primarily. For example, if you're interested in testing and developing solutions to reduce or inactivate zoonotic microorganisms, either in livestock or within the food chain. And I say myself and Karen Stevenson are here today and happy to discuss any potential projects with you. Thank you.
Okay, hello everybody. My name is John Lodge and I'm the head of the Food and Nutrition Research Group in Northumbria. So I'm just going to quickly summarise our capabilities. And hopefully you can see that would be quite useful to develop partnerships and collaboration with you. So we do work at both the food science level and also the nutrition level, and we also sort of do work more at the nutrition and food interface. Some areas that we run projects in are food processing and new product development, We have a small pilot plant, small-scale pilot plant, and we have the capacity to look at manufacturing, also recovery of natural functional ingredients from food. And these type of functional ingredients, could be bioactives for plants or compounds with structural or textural properties that we can take out, and recover from food, and then go on to the next level, which would be in terms of new product development. So we can take functional ingredients out and then create new products with those ingredients. So we're looking at also new product development, product process interactions, and especially in terms of what we're interested in quite a lot is in formulation of low GI foods. And this is headed by Dr. Nikos Loudis and Dr. Yorgos Koutsidis, who's in the audience today. We also have a lot of technology for looking at food diagnostics. So these can be bespoke, so we can do method development. We are very much interested in the characterization of nutrient and macronutrient composition, because we were looking at the nutritional value of food, what happens to the nutritional value of food during, for example, food processing, food manufacture, even home processing like cooking and storage in the home. So in terms of nutritional value, we're talking about the water of some of the vitamins are sort of an important aspect of food nutritional value which we'll cross through. Also, which is very important, is looking at the flavour profiles, compounds that involved in taste and flavour of the food. And related to that is the sensory attributes of the food also. And that's headed by Yorgos as well. We have benefited from a lot of research, a lot of infrastructure support, and we now have commissioned a multi-omics facility. So we're looking at doing proteomics, metabolomics, and genomic platforms specific for food research as well. So in terms of food security, we have the DNA sequencing, so we can look at the microbial organization in food and how that develops over time. We can also use metabolomics as a way to look at batch-to-batch differences in food, quality control, and also just to basically look at the changes to the composition of food in terms of biochemistry. So I'm a human nutritionist. We have facilities for human intervention studies, and we look at various clinical endpoints. Our colleague, Dr. Darren Smith, he's very much interested in types of bacteria which are growing in the gut. So we can look at, with the DNA sequencing, we look at gut colonization, microbial colonization, and the effect of diet on that. Dr. Trevor George is also a human nutritionist, and he looks at the effects of diet, especially fruit and vegetables, and bioactive containing fruit and vegetables. We look at that on vascular health, models of vascular health, and blood flow, and how the arteries and blood vessels dilate and relax properly. And he also has interest in satiety. Myself, I'm interested a lot in the diet and how the diet affects metabolism, using these novel approaches like metabolomics, where you can look at the whole metabolic component of the blood or biological samples and see how diet is affecting that and relating that to metabolic events. As well as that, we can also look at bioavailability, which is obviously very pertinent to the upcoming course. As a group, we have a wide range of food and nutrition research profiles at the moment. We have a good track record in terms of funding. We have very heavy workload in terms of industrial collaborations, all these with specific outcomes as well as just research profiles. We have a good record with TSB. Yorgos himself currently has 3 TSB projects just starting, and other research council funding. So what we're looking for really is partners and collaborations to develop, especially with the food industry, to develop projects specifically for these calls, opening calls. Thank you very much.
Hello everybody. I'm here to represent a large global company who highly active with RFID. Perhaps many of you haven't heard the name SmartTrack, but we are behind a lot of the global brands. If I mention Socumat, perhaps that's something that's familiar to you more than SmartTrack Technology Group. Essentially, we're an RFID manufacturer, high volume global production site, and we're looking for new opportunities and new partners for developing, possibly packaging, things like this that are becoming more and more important to prove where things have originated and where they're ultimately going to end up to add value. A nervous twitch has begun. Forgive me. So the company focus is basically innovative. We want to work with people who are perhaps putting together packaging, who want to add to that not just a simple barcode, but something that's a little bit more unique, something that can give non-line of sight data and a secure method of delivering information. With RFID, unlike the typical barcode, what you can have is encrypted, closed-loop data control that sits behind the RFID. So these are all I think, important things. You guys are all working on projects. Some of these projects will generate data that needs to be attached to specific products, whether they be living, animate or inanimate products. I think RFID ultimately might feature in some of your solutions. So as a global company, I think one of the earlier speakers mentioned that working with global companies with their own R&D it brings big advantages. SmartTrack has got 3,500 employees, and I represent the animal bio division. As a company, we've got significant experience across all use applications, from access control. Many of the passports that you guys own here today will have our technology in the back. So we are one of probably 4 companies globally that produce and manufacture the RFID inlays that fit in the back page. So credibility, I'm just trying to build some credibility to the SmartTrack brand by making these statements. Also, probably the biggest part of our business is the automotive industry. Basically, it's broken down into different sections. Each section is managed differently, but through the collective experience of all the individuals working all the use applications, you've got really good access to a great deal of knowledge. There's a lot of overlap, particularly when we start looking at secure systems. We've all perhaps had problems with the vehicle immobiliser, where we can't even operate our own car. That's because it's encrypted. We have the facility and the ability to encrypt data to specific RFID inlays that you might incorporate in some way into your data handling. So, I don't really know what more to say other than I'd like to obviously speak to you personally if you've got any interest. Exchange cards. I'm a lot better one-to-one rather than speaking en masse, so please forgive me. I'm happy to give out business cards, send brochures via email so that you know who we are and how we can possibly work together. Thank you.
Good morning to all. I am Anthony Caesar from University of Leeds. I am a Mercury International Fellow that is funded by European Union, currently based on University of Leeds. So our studies focus on improving the phosphate use efficiency in crop plants. As we know, phosphate is an important nutrient for life. It helps in several important processes of biological organisms, participates in the structure of DNA, RNA, biological membranes, helps in the regulation of key enzymes and so on. It is known that phosphate affects the production of crop plants on 60% of the arable land throughout the world. A recent Glasgow study conducted in our laboratory proved that it affects both growth and yield of oxide millet. Plants grown under red phosphate grow taller and produce healthy flowers compared to those grown under red phosphate. So it demands a supply of high input of phosphate fertilizers into the farmland. Where from this phosphate fertilizer comes? The world's cheap and inexpensive source of phosphate is obtained from the phosphate rock. So this is a major source for most of the phosphate industry. And unfortunately, this phosphate ore also contains a lot of selenium and fluoride, which is a major cause of concern for environmental pollution. And we cannot highly depend on this inexpensive phosphate for longer. Since the expected peak phosphate is going to occur around 2030, and all the world's inexpensive phosphate reserve may disappear in next 100 years. And another problem is that it is estimated that farmers often apply 4 times higher phosphate than that is removed by the harvest. The runoff of excess phosphate from the farmland to the ponds and lakes cause the algal bloom, a major concern of eutrophication. So with this background, understanding the basic mechanism by which plants uptake the phosphate from the soil and redistribute inside the plant will definitely help us to improve the phosphate use efficiency and also reduce the input of fertilizer to the farmland. If we take the concentration of phosphate in soil, it seldom exceeds 10 micromole, which is 100-fold lesser than that of cytosolic phosphate. So plants possess some efficient transporters in the root hairs for the accumulation of phosphate from the soil. Some plants also could be able to form symbiotic association with mycorrhiza, which also helps in the accumulation of rice phosphate. So these are the high affinity phosphate transporters available on the membrane. So these are hydrogen phosphate symporters, transport both hydrogen and phosphate with the expense of ATPase, through ATPase mechanism, which is well established. So these are a group of genes. Now they are found throughout the plant from shoot apex to root apex, involving both in accumulation as well as redistribution of phosphate within the plant. Their transport properties and affinities are characterized by expressing in heterologous systems like yeast and xenopus oocytes. Spatial expression and structural features are analyzed through molecular and proteomic approaches. These background studies would help to improve the crop plants through transgenic modification or marker-assisted breeding, which could help reduce the input of phosphate fertilizer and offer a cost-effective and sustainable agriculture. So in our university, we have a rare combination of 3 different expertise: Professor Elsenbecker from plant science, Steve Baldwin, membrane transport biologist, who works several decades characterizing many membrane proteins, and Steve Munch is a structural biologist recently joined our group. Thank you.
I just wanted to tell you a little bit about Agri-Food and Biosciences Institute, or AFBI as we call it for short, to see whether you're interested in any of the research and services that we can do. AFBI itself has about 850 employees, and we carry out research and services right across the food chain, soils, aquatics, plants, animals, right through onto the food side, biometrics, economics, the whole lot. The food science is a combination of food microbiology and food chemistry. The 2 main areas that we're concerned with are food quality and food safety. Food quality, I've just identified a few key areas again within that. On the nutrition and quality side, I'm involved with a lot of work on the fatty acid side. Especially with meat, milk and eggs, both on farm and in the factory, to manipulate or to optimise. We've then progressed looking at other micronutrients such as vitamins and antioxidants, and how animal production systems can be managed to optimise the quantities there. And in fact, we're doing quite an unusual study at the minute where we're looking at UVB light and vitamin D in milk. So we can be quite inventive when it comes to the point. Eating quality and novel processing techniques and minimising food spoilage, these are all, if you like, supporting all the research that goes on. On the eating quality side, we're now moving on to online sensory assessment of foods. We have trained, experienced and naive panels to work with, and we use both instrumental and other chemical analysis to look at the attributes that the consumer is interested in. The novel processing techniques, some of you may be familiar with Margaret Patterson. She has done quite a bit of work on high pressure processing, and she's now linking with other colleagues here on the microwave technology side. On the safety side, again, it's the 2 areas coming together, the microbiology and the chemistry. We have facilities to look at pathogens, to do challenge testing. We're interested in the microbiological status of food processing plants, going back to slaughterhouses, going right forward to packaging areas. And we complement that with the analysis of food contaminants such as pesticides, heavy metals, that like in foods. We've been just finished a project for the Food Standards Agency where we've been looking at the fate of pesticides in fruit and vegetables. If you want to discuss any of this further with us, I'm here, my colleague Mark Linton is here, and I have some handouts and some cards with me as well. Thank you.
Sooner. Yeah, okay. So, Food and Health Innovation Service. I'm pleased to say that 85% of what's in the scope document falls within what Food and Health Innovation Service does in Scotland. And really, Food and Health Innovation Service is going to enable companies in this room, but more importantly with the academic partners in this room, the companies that are in your consortium for Nutrition for Life call, we can work with them. And I'll tell you how we do that and why that's important. The F stands for fun as well, I'm sure. The Scottish Enterprise will be pleased that we might change the name to Fun Health and Innovation Service. But it's a 5-year project. We're in year 3 of the project, and it just gives you some idea of the market for the food health market opportunity, both in the UK and globally. What's important is that the consortium is led by Camden BRI, who I'm sure you've all heard of, and our 3 other partners, 2 of which are in this room. Fiona's here to answer any questions over lunch as well. But it's a unique combination of partners that can support companies both from a technology, health science and food marketing for health as well. And I think that's what's really important is in terms of what we've heard earlier, is that we're going to be able to work with companies to help them to understand the market intelligence for the products that you want to develop, and also marketing for health, which is quite a specialty. And I think that covers the C of the ERICA in terms of getting that commercial proposition correct in the proposal that you're going to do to the TSB. So there's our partners. It's for food and drink companies in Scotland who may be wanting to reformulate products, develop new functional food products, or exploit naturally healthy food and drink. And it is unique. It's a comprehensive resource, and essentially what's going to, I'm not very good at this, is work on a one-to-one basis with the companies in your consortium, both in terms of, there we go, the slides, yep. Yeah, not really the slides. But anyway, what we're able to give with companies is through a one-to-one meeting, identify where their gaps are perhaps in the proposal that you're doing for Nutrition for Life. Being able to give them the information that will make your proposal more robust in terms of marketing, the regulatory framework for health enhancing, for foods with a health benefit, but also the technology and the processing side as well. Here's our 6 themes for 2013, and I've set against that 4 of the key themes in this document. So you can see where there's a synergy between what we do and the call that we're talking about today. So companies that want to come with us, we can identify with them where they need market support, and that might be through market intelligence, it might be through marketing for health, but also the regulatory framework, which we spoke about earlier in terms of making that case. We can also provide scoped up technical approaches to what the company wants to do with you through the application as well. We can leverage in the technical expertise from the Camden, through the Scottish research organisations and university, and in particular working with the Rowett Institute for Nutrition and Health in terms of the emerging health science. So what we can do, have a look at our website. Even if you don't want to come to us, we've got amazing resources on our website for different sectors, but also in terms of the themes. So some of the documents that there might actually help you to write your proposal as well and give that market evidence that the TSB will be looking for. So, quite a few case studies now available from the companies that we've actually worked with, both in terms of their journey to date and in terms of products that we've helped them to develop as well. We've worked with over 140 companies to date on a one-to-one basis, and we have over 350 companies in our community. So we're an excellent resource that hopefully we'll be able to help you with. Now, I was interested in the deadlines, so noticing that the final deadlines for this call, if you as a company or the companies in your consortium come to us now, we can have that one-to-one meeting arranged with them in April and May, and within 6 weeks type of timeline, we'll be able to deliver our deliverables to the company in terms of marketing support, technical support. So that is well within the framework that you're working to. So I can see that we can obviously be of some help to you. People like us, if I meet you, we're good fun as well as being excellent at what we do. And as I say, Fiona and I are in the room to take questions, and obviously that's my time up. So thank you very much for your attention.
Elizabeth Crampton, and I work for a company called EWOS, and our business is to produce salmon feed. And you might have noticed salmon on the menu earlier for lunch. I hope you all managed to enjoy it. It was delicious. And we produce, as I just said, we produce a lot of salmon feed, and that's globally. That includes the UK, Norway, Chile, Canada. And although not salmon in Vietnam, we've just started feed production in that country. We have a sister company that produces salmon itself, and we have the growth in feed volumes has been continuing for many years now. And although feed volumes have grown, the amount of raw materials from marine sources is stable and even declining. And that means that the inclusion as a percent of our feeds, the inclusion of marine ingredients, that's fish meal and fish oil, they have been declining. And for instance, we now make our feeds with more protein coming from plants than we do from fish. So although salmon aren't yet vegetarian, the ones we grow are not truly carnivorous anymore. Not by choice, probably. But that has some knock-on consequences. And the main knock-on consequence is that there are nutrients that are present in those marine ingredients which are absent or low in plant proteins. We're talking about long-chain polyunsaturated fatty acids, vitamin D, selenium, and would like to try and address some of that. So, what we'd like to be able to do is increase the level of those nutrients, those beneficial nutrients in the fish that we produce. And of course, as a feed producer, we're part of that human food chain. Our fish is what they eat, and you are what you eat. And if you eat salmon, it follows that you are made up partly of the feed that we make. So, although you might not like to dwell on that for. Anyway, we have a research company, EWOS Innovation, and we have a lot of trial facilities. We can grow lots of fish in lots of different tanks. So we could, for instance, have different levels of vitamin D in the feed and monitor and calculate how much of that vitamin D is retained in the fish muscle. We have pilot-scale feed production to make those experimental lots of feed. We can do routine fish analysis and feed analysis, but we can't do specialist raw materials, specialist analysis of raw materials or fish or feed. So for instance, if you're skilled in vitamin D analysis, then we'd like to speak to you. And any other nutrients that you feel are lacking in humans, those are the sorts of things we usefully collaborate on. And we'd like also to improve our understanding of some of these mechanisms rather than just feeding the whale. So if any of those things ring any bells, please contact me and we'll have a chat. Thank you for your attention.
Hello everybody, my name is Marion Smith. I'm business development at Queen Margaret University, so apologies first, I'm not a scientist. I'm going to be talking in very kind of broad terms about what our particular interests are and also our expertise. So Queen Margaret University, we're a small university, we're based just out at Musselburgh, and we're particularly interested on the food, health and diet side of things. One of the things that we really are looking into at the moment is food security, and I understand that this isn't a term that chimes particularly with businesses. So from my understanding, it's about the sort of economical sustainability of the food supply chain. It's about improving and maintaining the nutritional content of food and also sort of optimising nutritious and affordable food. And one of the projects that we've been doing with a small business is about putting a larger company in contact with them who produces marine algae, and it's about putting it into dressings and oils, and I think that's a really good way of sort of addressing both the economical sustainability of the food chain and also improving the nutritional content of food in a way that people might actually not even realise that they're getting the omega-3 and the fatty acids into foods. So it's it's a very interesting project which we're actually doing with a company based in Scotland. We're also looking at naturally healthy foods. We're very interested in sort of development of functional foods, specifically in connection with diet and health again. And we're very interested in anything to do with sort of better for you products in terms of the reducing of fat, sugar and salt content. We're working with a larger company who's over in the States about looking at reducing salt content in bread as one of the projects we're doing as well. So from my point of view, as I say, I'm not a scientist, but our sort of underpinning research is in nutrition and metabolism and diet and health. That's the underpinning research at QMU, and we have a range of dietitians, we have a range of staff with biological sciences backgrounds, we've got nutritionists, all of whom are very keen to work with businesses, and we're working with a range of, as I say, small businesses and large businesses. Our expertise is really in developing kind of new and innovative ingredients. We're looking at, as I say, replacement ingredients and anything to do with the health enhancing ingredients in terms of because that's where we're very interested in, particularly in diet, and diet as it affects health and disease. It's one of our kind of team at the university is Professor Isabel Davidson, who heads up, and she's very interested in sort of diet and specifically into the ageing population. Other of our team is Jane McKenzie, who has an area of expertise in omega-3 and oily fish side of things. Elaine Bannerman has expertise in the sort of gluten-free side. So as I say, we've got a whole range of expertise. We're very interested, as I said, in new ways, approaches to nutrition, as I've said before, with reformulation, innovative ways to improve nutrition intake, as I've said in the marine algae example, and also working with novel processing techniques. We've had a very good partnership with AMT, where you've heard Sue talk about what we've done with salt reduction, and we're very interested in anything like that. So that's me, and thank you very much.
So my name is Grant Seller. I'm one of the business development executives at Heriot Watt University. So Heriot-Watt is a very applied university. I was reminded at lunchtime that, of course, the Sunday Times Scottish University of the Year 2 years in a row. So we have some good recognition in the community. So I'm going to talk a little bit about kind of food protein functionality and a couple of areas at Heriot-Watt that I hope may be of interest in this call. So our protein functionality group, particularly within the School of Life Sciences at Heriot-Watt, spans a spectrum, an interrelated spectrum of structure, functionality and sensory aspects of food and drink. So under our expertise in the structure-related aspects, focus around particle size distributions, a very kind of basic kind of aspect with particle size distribution, confocal microscopy facilities, various kind of modelling techniques, and we also have a proteomics facility which does multi-time of flight mass spec analysis. We're moving on to functionality aspects of food. We have expertise around the areas of foaming properties, not just in food stuffs, but also our International Centre of Brewing and Distilling looks at foaming properties in beer. So it's a very nice place to work, since we have a pilot brewery and a pilot distillery around the corner from my office. We also look at emulsification, rheology techniques, so looking at gelation, thickening, etc., and we have model systems around those. And then connected with that, we have a sensory suite on campus. So our campus is based at Riccarton, just outside Edinburgh, the outskirts of Edinburgh, quite near the airport. So we have, in the sensory suite, we can look at kind of flavour, colour of foodstuffs. We have a trained panel available. We have specialised statistical correlative expertise as well, and we're involved in food model kind of testing. But as I said, Heriot-Watt is a very applied university, so we have a lot of history of working with industry and applying these good techniques into industry-related problems and addressing those issues. So some of our kind of current projects are really around kind of fat replacement and novel fat-free products. In fact, we've had a TSB project from the previous Nutrition for Life call, Nutrition for Life 1, working with a very large food manufacturer in the UK. It's a multi-collaborative kind of project. Some of our other projects are around kind of enhancement of functional kind of properties, looking at the next generation of functional properties, and also egg protein replacement for an active area. We're also looking at the valorization of food waste streams, so things like extraction of food-grade proteins from waste streams or kind of co-products from the brewing and distilling industry, extraction of novel proteins, including ones with antimicrobial functionality, and also looking at enhanced foaming properties, such as kind of surfactant proteins. One of our other areas is actually Dr. Mihalis Panagiotis, who's a recent kind of, recruit to Heriot-Watt, who brings a lot of expertise in natural products and the bioactive properties, and also benefits in health and disease. So his focus is really around the determination of biological activities of these products, so antioxidant, antimutagenic, and very importantly, anti-cancer properties. And at a more kind of basic level, this relates to the determination of gene targets, underlying mechanisms in cell death pathways, for those who are interested at the kind of cellular level of these aspects, oxidative stress pathways, epigenetic pathways. We heard a little bit about epigenetic pathways this morning. So Mihalis has a number of current industry academia projects running, a couple of particularly of interest around novel functional foods containing bioactive essential oils. And these are from pre-pandemic species with health-promoting properties. Another one is a bee-based natural product, and that's looking at as a potential cosmeceutical agent with health-promoting properties against UV-induced skin photoaging. So something which is very relevant in our changing climate. So my name is Grant Seller. I'm a member of the business development team. My contact details are there, and we'd be very happy to talk either today or outside of the events about the possible collaborations. Thank you.
Welcome everybody, I'm Andrew Whitley from Bread Matters. I'm a bit of an oddball here. I don't have any status like you people do, academic or business or otherwise. My CV, sort of brief CV, gives you some sense of what I've done, but it doesn't say that I started life as a Russian specialist and broadcasting in the BBC to the Soviet Union. And there's a saying in Russian, among many sayings about bread, which is "Хлеб всему голова." It all starts with bread. And of course I would say that, wouldn't I? But actually when you think about it, bread is a key foodstuff, and the raw materials for bread are vital in so many ways in our diet. And my proposition here is that we need to do something to fix not just the food system in general, but the bread system, which is interleaved with it so radically. Because it's not really doing what we need it to do at the moment. It's based on a rather linear productivist model, which suggests that the function of the plant breeder is to produce bigger yields and more functional effect in the grains that we make into bread. The baker is to puff it up with additives to make it look even bigger, and the consumer is to put up with the fact that they can't make head or tail of the ingredients that go into bread because they don't understand the names, or indeed that they aren't actually mentioned on the label, because guess what? They don't have to be, because the law enables bakers to hide certain things like enzymes which are used in production. And the net result is we've got a massive problem of diet-related ill health. Where it relates to bread, it's expressed in the long-term per capita decline in bread eating, because many people have experienced bloating, irritable bowel syndrome and various other things when they consume industrial loads. And we have other causes of significant problems like obesity and coronary heart disease, which are not contributed to at all by the current state of the bread that is eaten by most people. Food sovereignty is a term I prefer to use than food security. Security implies that you just need to assure that you've got enough money to buy the food wherever it's produced. Sovereignty implies that we need to take some control over how and where it's produced, and where relevant, to bring it a little closer in the interests of reducing its carbon impact and reinforcing, sometimes recreating, the connection between food producer and consumer, which is the basis of what we might gradually work towards understanding as a sustainable diet. So if you are at the end of one of these extended modern supply chains, you're subject, as the wheat producers and millers are very much at the moment, and hence the bakers, to the nasty activities of the food speculators, the financial people who will use any opportunity to insert their power into anywhere where trading goes on with commodity products. And part of my aspiration and what I'm pitching to you today is an attempt to say we could do this differently if we all got together using all our expertise, from the plant breeders who could be invited, for instance, to look not just at yield and at functional effect, but guess what? At digestibility and nutrient density, characteristics of grain that are only ever measured when we come to feed them to animals. It's a scandal that nobody's aware of, except in specialist circles, that we don't even ask ourselves what the wheat that farmers are planting and growing for human consumption have in them other than protein and functional ingredients like glutenin and gliadin and so on to make puffy bread. So we've got serious work to do there about defining what might be a better quality grain. And then we need to ask ourselves why millers are milling the hell out of our grain, leaving nothing useful in it, and taking all the useful ingredients and either selling them to farmers who are well aware of what is good to feed to their animals, rather than to leave more into the grain. So my proposition is that in the view of the fact that the current research tends to be very much in support of the prevailing paradigm, which I would suggest is jiggered, we should go for a new research system, if you like, which involves participation by all people who have an interest in the process, from soil scientists right through to ordinary citizens. We need, the aim being, as it says, a Scottish bread supply which is healthy, equitable, locally controlled and sustainable. And this means looking at each segment of the process and engaging the best brains and the best research and the best evidence, putting it all together and saying we could do this a whole lot better. And the net result would be not just fun, as was mentioned by a previous chair of these sessions, but actually what this is all about, which isn't just a product, although there will be bread products and other baked things resulting from this, but I would suggest health, which is really what we're all about. Nutrition for life is not just about producing products by businesses, it's about having a healthy and happy community. Thank you very much. And by the way,
yes, David Kennedy. I'm from the Brain Performance and Nutrition Research Centre at Northumbria University. And just to give a rough idea of what we do GCP quality control trials in humans, and we look at basically two things. We look at brain function, so go back there. So brain function, which is sort of on the left-hand side there. So we mainly look at cognitive function. We look at brain imaging, cerebral blood flow, mood, psychological state, etc., and various sort of analytes that we can take from blood and other bodily sort of fluids in order to gauge sort of brain function. We also look at physical performance, so in particular, obviously, just actual physical performance, but we look at appetite, satiety, different sort of physical sort of parameters and what have you. And we look at these with respect to foods, food supplements, components of food, individual components, micronutrients, right the way down to single ingredients that, say, a large manufacturer might be interested in introducing into a product as a functional sort of ingredient. So those are mainly sort of cognitive function, physical performance, anything to do with food. And just to give an idea of some of the type of research that we typically do, I've just picked out a few different publications, and you can find the rest on our sort of website there. So, for instance, on the brain function side, we've done some recent work on resveratrol and polyphenols. We've got the most, I think, probably the first studies sort of showing brain function effects of resveratrol, which increases cerebral blood flow, flavonoids. Quite a few studies looking at vitamins, using multivitamins rather than sort of single vitamins. A lot of work on caffeine, omega-3 fatty acids, showing changes in cerebral blood flow and cognitive function. And then on the physical performance side, we're doing a lot of work at the moment looking at dairy products with regards to physical performance, satiety, appetite and muscle damage, so into preventing muscle damage. Again, calcium, glycemic index and sort of caffeine. And that's it. That's sort of what we do. Thanks very much.
My name is Musharraf Sarkar. I'm from Teesside University, and my interest, actually, inflammation, the mechanism, the way the inflammatory mediators regulate our body functions. My main research career I developed at King's College London using animal studies. And for personal reason, I moved to Teesside University, where I was warned that you can't use animal there. So I have to switch on something using human tissue. So I started using my knowledge to understand about inflammation again. But at that time, one of my friends, who is a consultant physician at Chicago University, introduced me this bacteria, pro-beneficial bacteria. I didn't have any understanding what are those. I know we eat yogurt and they are beneficial bacteria, so we need beneficial bacteria. And interestingly, people although knew the beneficial effect of bacteria from ancient time, but nobody actually bothered to look at those, why we need this beneficial, how they're interacting with us. And it is only probably from 2000 or 2001, all the publications pouring in, showing that they are really important. Even Nature, over the last 5 years, published few papers showing that the animal, if the animal, they don't have probiotic bacteria, their brain becomes small, heart, short, liver, small, kidney, develop smaller size. So you can imagine the way they interact with our body. Even the problem with current, gastrointestinal disease like inflammatory bowel disease, because those people actually has mutated gene, and this mutated gene, they actually produce a kind of immune cells. When that immune cells interact with gut pathogen, they become activated, and they can go back. They stay activated, release bad chemicals, and that causes diarrhea, pain, gastrointestinal discomfort, all sorts of things. But recently, a lot of clinicians, even 4 years ago, if you ask, treat your patient or give some probiotic, that will improve, they will say, you're mad. They like antibiotic. But even now, people from Harvard University, Chicago University, they're treating their IBS patient with probiotic, this beneficial bacteria. So I developed my interest, how this beneficial bacteria are talking to us. Let's do some real research. So I started growing this bacteria in my lab because there are a few hundred times. Rather than working on every single bacteria, I took one bacteria which is very well studied, very well known, and grew them in the lab, then extracted the media, not the bacteria itself, then interact with macrophages, the immune cells. And surprisingly, we found that this bacteria can modulate physiological function of macrophage. The macrophage, they actually roam around our body, look for bacteria, virus, or dead tissue, and they eat them and destroy them. So I just allow this macrophage to graze on a pathogenic bacteria, E. coli. Then we looked at the rate of digestion of that bacteria. And you'll be surprised that the probiotic, they increase the rate of digestion is so much, where this bacteria, when I activated with pathogenic bacteria, it produced lots of bad chemicals, but they didn't do anything about the digestion, the behavior. They didn't change the behavior of macrophage. But the probiotic, in presence of the probiotic bacterial media, the macrophage is totally activated and start digesting this bad bacteria faster. Then I looked at what happened to the chemicals that cause the problem, that cause the IBS or gastrointestinal dysfunctions. And one of the chemicals is nitric oxide, pathogenic nitric oxide. When I introduced this nitric oxide into this macrophage, they produce huge amount of nitric oxide, which is very inconsistent with the findings from the patient with problem with gastrointestinal diseases. When I applied those chemicals in presence of probiotic bacteria in a different concentration, you can see they produce nitric oxide, but they have cut-off effect. They reduce the amount of production of nitric oxide as a dose-dependent manner. By themselves, they actually produce little bit nitric oxide, but those are probably more physiological, maintaining this, because nitric oxide you need to maintain your health. And we established that whether, this is a very interesting findings, and we use different methods like fluorescence microscopy, molecular technique, and also other signaling molecules to explain those mechanisms. So what I'm trying at TISA University, trying to understand the specific bacteria, probiotic bacteria, that way actually interact our body, trying to understand their mechanism.
Clark, and I work at the University of Aberdeen. I'm a food scientist. Unusual species though, because prior to my academic role, I worked for the food industry. And prior to that, I worked for a research institute. So I cover all various sectors that we've seen here today. There are two aspects that I would like to speak about. First of all, our NO, secondly, about the team. But before that, a project that we are, this is, okay, let's start with that. A project that is a collaboration with the James Hutton Institute, which involves the utilization of novel food ingredients. In this case, we looked at the properties of flour coming from fava beans. And the idea is that fava beans are very beneficial for the environment. very beneficial for agriculture because it can utilize this feed. And with this project, we utilized the byproduct of the animal feed industry for the manufacture of breads. And this slide actually shows the capabilities that we have in Delegate. Using experimental designs, we optimized the composition of a bread that contains wheat flour and whole grain flour. We looked into the texture utilizing CT scanning and connected the neurological properties of the bread as we studied them with an empirical instrument, texture analyzer, and connected this behavior with the images that we get for porosity and connectivity of pores as obtained by the X-ray computerized tomography. We believe, myself and my colleagues in James Hatton, that fava flours offer a huge range of functional properties that they are not well known or explored. So we are looking for partners who are interested in the development of functional ingredients coming out of these flours, or the development of new functional novel foods. For example, the fava flour breads have got a higher calcium, higher zinc, higher magnesium contents than traditional wheat bread. And I think with this gentleman who spoke about the bread, we have to readdress what is the role of bread in human nutrition and health. So we'll be looking for partners that perhaps can help with the characterization of the starch and protein fraction in the power plant, pilot plant facilities, and perhaps snack manufacturers. And now the team, University of Aberdeen has got a technology platform called Food Innovation Aberdeen, to whom I provide support as a scientific advisor and along other members. My background is in rheology and texture analysis, interactions between milk proteins and food hydrocolloids, and interactions between food processes versus food ingredients. What combination of food ingredients will give the best result for the food process. Background in sensory evaluation, and now I'm involved with X-ray computerized tomography. Other members of the staff apply ultrasonication sonochemistry to promote functionality in foods. We have Catherine Chang, who is involved in antioxidants in foods. Adelia Limus is also in antioxidants and novel preservation methods. Graham Walker is an expert in brewing. Jonathan Wilking is snacks and lipid oxidation. Maxine Clark is in reformulation and product development. We are also having a new course in packaging, so we hope in the future we will be able to help customers in problems with packaging. Thank you.
Mike McInnes, I'm a retired pharmacist, talking about a product which has got honey and a particular amino acid dipeptide in it. The human brain is shrinking for the first time in our evolutionary history. The human body is expanding at the expense of the brain. Humans are becoming demented. More than 1 billion humans will be demented by the year 2100. Homo sapiens is fast becoming Homo insapiens. Consumption of energy-dense, refined carbohydrates and sugars, new in human food culture, overloads the systemic system with energy and triggers the cerebral trip switch, the glutamate-glutamine cycle, responsible for bringing glucose into the brain. This short-circuits the brain. Glucose entry is blocked and leads to chronic cerebral hunger. More high-energy food is consumed with the same result. And the cycle repeats again and again and again. The excess energy in the systemic system is converted to fat, leading to obesity, metabolic syndrome and dementias. This may be resolved by achieving nocturnal energy homeostasis prior to sleep. This is open to every human being. The holy grail of human metabolic impairments, as used to happen in the southern Mediterranean. They replenish the liver prior to sleep with a late healthy evening meal, 10 to 11 p.m. Forward provision the brain, activating sleep and recovery physiology, reducing chronic nocturnal metabolic stress and risk of metabolic syndrome. Since they have abandoned this practice, the fastest growing region for metabolic syndrome, that is obesity, diabetes, heart disease and Alzheimer's disease, is in the southern Mediterranean. Honey, as it happens, is the gold standard food for selective liver replenishment prior to sleep, after an early evening meal, as we do in the West, without digestive burden. Honey partitions glucose to the liver by several mechanisms. Honey is a potent anti-diabetic fruit, actually the most potent known to man. Stabilizes blood glucose, reduces both hyperglycemia and hyperinsulinism, the twin toxic hypers that suppress the cerebral glucose pump and block glucose entry into the brain. Contrary to popular opinion, sleep is a high energy system with respect to the brain. Honey activates the honey insulin melatonin cycle, the HIM cycle, and promotes a cascade of homeostatic hormones. These are GLP-1 glucagon-like peptide 1, free IGF-1, leptin and melatonin, each of which individually and synergistically recalibrates the cerebral glucose pump. Okay, this is the key. The lovely dipeptide, I won't tell you the name of it, 2 amino acids, natural to human physiology, synergizes beautifully with honey. This dipeptide is a potent promoter of glucagon-like peptide 1, or GLP-1. The most potent and positive influence in human metabolism. This GLP-1 has positive influence in the gut, the liver, the circulation, the heart, and the pancreas, and the brain. And it is a powerful suppressor of hyperglycemia, and of appetite. It's emerging as a really, really significant new, and we can promote it every night in life by doing that. We're protected this way. The patent examining officer has allowed the combination for insomnia, sleep apnea, and sleep problems, and described it as novel and not obvious. 8 hours of quality sleep and recovery physiology will burn 5 times the body fat compared to a 90-minute workout. I'm going to repeat that, because very few people know that. The only scientist in the world that is saying this openly is Robert Lustig. 8 hours of quality sleep and recovery physiology will burn 5 times the body fat compared to a 90-minute workout. The IP, which is 15 years of research, is embodied in the patent. The product will consist of a spray-dried soluble powder for incorporation in food and beverages. The University of West of Scotland are currently engaged in phase 1 study funded by Scottish Enterprise. The retail cost will be around $1 per day. This may be the most significant and cost-effective contribution to public health and learning in several generations. That's open to every single person that can afford the product, which would just be about $1. Let me give you a very simple example of how this can affect you. The last meal of the day in UK hospitals is 6 p.m. That means that nobody in any hospital bed is recovering optimally. All they need to do is replenish their liver selectively before they go to sleep. Thank you.